As all of you, I hope, know, the class on Wednesday was canceled. I hope you didn't try to get here. And that means that it's necessary to tweak our schedule to take that into account. And I think what I've come up with is something that should accommodate it without causing too much chaos down the line. This is a revised schedule for the time between now and the second hour exam. A copy of it is available online at ICON, and I'll show you that in a moment. But the key things to be aware of are that on Monday we'll be doing Chapter 5, which was scheduled for today. And then the first hour exam will still be held on Friday the 15th, but it will only cover Chapters 1 through 5. So you'll still have time between Monday and Friday to incorporate and review and revise and so on. The second hour exam is still going to be left to the Wednesday the 12th, and we'll cover all of Chapters 6 through 10. And what I've done, if you're interested to compare it to the previous schedule, is I'm only accounting for 2 days for Chapter 10, which has in the past been kind of a big chunk. And it may end up more being 2.5 and 1.5, somewhere in there, but that's where that extra day is being trimmed from the semester schedule. And then we'll be back on the previous one as of the second hour exam. Does anybody have any questions about that or concerns? Let me just escape from that. And I'll show you where that is on ICON, so you don't need to worry about getting a copy. If you look here, on the front page of the course is an attachment where you can download that schedule for yourself if you want to do that. I'd also like to remind you that both Lynn and Tim are available to meet with you. The times are prominently front page there on ICON. So Monday, Tuesday, Wednesday, there are chances to meet with the TAs. If these times don't work out to be the best ones, Tim, do I have your times right there? Is that correct? Okay. If these turn out not to be the best ones, we can change the schedule during the semester, but give it a good try. Also on ICON, if you want to go to the content page, I'd like to draw your attention to Genome 2.0 and Ode to the Code are supplementary materials, not required, supplementary materials on the DNA. The protein poster here is the very elegant picture I held up and waved around so you can have your own electronic version of it. And some other papers that will be more relevant later on. I'm curious to see how this works, and it, okay, let's try that. All right. I want to clarify something. Someone was asking about GC pairs and why GC-rich DNA is more stable against melting. And I want to make sure that it's clear in everybody's minds because it's easy to slip over into something. It is true that GC pairs have 3 hydrogen bonds compared to the 2 of an AT pair. It is true that GC-rich DNA melts at a higher temperature. But it is not because the hydrogen bonds make the helix more stable. And if you remember, and I'll go back to the original PowerPoint to point it out to you. In fact, I think I'll just do that right now. No, I have that one already. The DNA, here we go. If you happen to have with you your DNA handout still, the figure 20 on that one, it's the wrong chapter. That should be chapter 3, sorry. But it's slide 20. Page up. Okay. Slide 19 in your DNA handout. The stability of the double helix is due to these things called stacking interactions. The aromatic groups tend to align in the plane with each other. There are weak stacking interactions between the nucleotides in solution. But when you have a polynucleotide, like the nucleic acids, those stacking interactions get much more important. But the exact strength of the stacking interactions depends on which bases, and it happens to be the case that GC base pairs have stronger stacking interactions than the AT ones, not because of the hydrogen bonds. And you notice it says here, GC stack more tightly. And it's just, so we did make the point in lecture that the melting temperature is due to the stacking interactions. The 3 hydrogen bonds between the GC pairs are important for having specificity, for having the right helix lining up with the right helix, but they are not what accounts for the fact that GC-rich DNA melts at a higher temperature. Are there any other questions about that? Because someone had asked me about that. Okay. So, any other questions about course organization or about the material we've talked about so far in the proteins? Okay, so let's get on with proteins. All right. So we had, okay. So we've talked about the idea that linear polymers like DNA and protein have a primary structure, which is simply their linear sequence. We've talked about how for the proteins there's secondary structure of either the alpha helix or the beta pleated sheet. And we've talked about the concept that tertiary structure is the overall packing in space of all the parts of a single polypeptide. So how does a protein fold into that one particular tertiary structure? And it turns out that the major driving force for proteins is the hydrophobic effect, the fact that nonpolar residues tend not to interact with water strongly and therefore end up apart from water together, like the unpopular people at a high school prom. They all end up in the corner together. See, it'll stick in your mind forever now, right? There are hydrogen bonds going on. There are hydrogen bonds in the alpha helix. There are hydrogen bonds between side chains in some cases. But because those hydrogen bonds would have been made to water before they were made inside the tertiary structure, the hydrogen bonds can contribute the specificity of the structure, but they don't contribute to the stability of it. It turns out that the disulfide bridges, SS bridges, aren't the thing that drive it into a folded state. Those are formed after they are completely folded. And the ion pairs, in some cases within a protein, you'll see a positive and a negative charge addressing each other, across an interface or something. Those would have been interacting with the water if they weren't interacting with each other. So again, it contributes specificity, but not necessarily stability. In contrast to that, we can see that the zinc actually does stabilize the zinc fingers to some extent. So we're going to see later on when we look at proteins that interact with DNA, regions called zinc fingers. And the zinc in that case is important for stability. And if you took that zinc out, it would tend to unfold. These are a couple of sort of schematics for you, if you like, comparing unfolded or misfolded proteins to globular ones. They're among the few that I've been able to find that aren't in red-green. So here you can sort of schematically see the notion that as a linear molecule, it could flop around in a variety of ways, or it could have its hydrophobic parts mismatched and come up in an inappropriate one. Proteins that are either unfolded or misfolded tend to stick to each other because those hydrophobic regions are still hydrophobic. They tend to be easily digested by proteases because their peptide bonds are out flopping in the breeze. They tend to bind to a special class of proteins called chaperones. And in the last few years, we've discovered that there are proteins that can assist other proteins to fold. And in general, as long as a protein is unfolded or has been misfolded, it won't be able to accomplish its proper function. If it doesn't have the proper structure at the tertiary level, it won't be able to function. By contrast, if we look then at a globular protein or a properly packed one, especially the globular ones usually are soluble as opposed to aggregated. They will be less susceptible. They won't be completely resistant, but they'll be less susceptible to the proteases. Once they're completely folded, they're not going to stay bound to a chaperone. And it's in that tertiary structure, the fully folded structure, that you're going to actually find a functional protein. Now, if you think about it, this is pretty amazing. I mean, if you think about a protein that's got 40 residues or 80 residues, how many different possible ways of folding that thing up might there be? It boggles the mind. It's been one of the major problems for biochemists for many, many years. And this is giving you an example of one of the ways of thinking about it that's been fairly fruitful so far. If we start with a protein that is in its correctly folded state, and this particular one is one that has disulfide bridges, ones that has SS bridges to it. This one can be detected as properly folded because it's going to be catalytically active. It'll have some enzymatic activity. We can make this unfold by adding urea to disrupt the structure of the water and mercaptoethanol that will break those disulfide bridges and end up with an unfolded state like this where we have now sulfhydryls again instead of the disulfides. If we remove the urea and the mercaptoethanol, this spaghetti pile will, for the correctly chosen protein and conditions, fold back up into something that can be shown to be the same as it started out as. And the suggestion has been made that there's like a funnel of folding that's going on. That in the earliest stages, what you're looking at is just little local bits of order forming, little local bits of secondary structure. But that as that happens, there are fewer states that have to be explored. And partway through this folding process, you have what's been called the molten globule. The idea that there's a fair bit of secondary structure that's correct, but the hydrophobic residues are just kind of milling around in a not very well organized state and still pretty mobile relative to their final state. But they're starting to withdraw from water so they can start to kind of elbow around and try to find that minimum packing. And then from there, you can end up with sometimes actual intermediates in folding down to the final structure. So there's a... It's worth keeping in mind that protein folding can actually start while the protein is being synthesized. If you look at the rates of protein folding and the rates of protein synthesis, we can predict that at least some proteins should start folding as partial peptides or nascent peptides. And this is, we've mentioned this before. You can imagine that there might be ion pairs between things like lysine, which has a positive charge, and glutamate with its negative charge, or arginine and aspartate. And these are actual ion pairs that are found in myoglobin. But again, because their charges would be interacting with water if they weren't interacting with each other, they don't direct the... I think I actually have that backwards. I think I would want to put that around the other way. They don't direct folding in the sense that they are in charge of it or that they act to catalyze it or anything, but they do help select the final correct structure. Disulfide bonds are not very common inside the cell. The cell has a reducing state, if you like, compared to the outside of the cell, which is slightly oxidized. The thought is those disulfide bridges may help to stabilize extracellular proteins in a somewhat harsher environment. We do know that some of the proteins that have disulfide bridges can still function as enzymes even after those disulfide bridges are broken, as long as the secondary and tertiary structures are still there. And then this is just a picture to show you what I was talking about in terms of the zinc finger. Zinc is a very complicated little atom, and it can interact with multiple things at once. So it can have anywhere from 4 to 6 things that it's interacting with as an ion. And that allows it to sort of act like a centerpiece to the bouquet, if you like, to hold those things in. And we're going to see that zinc domains are common in DNA-binding proteins. So protein folding, and therefore tertiary structure, is a consequence of the primary structure or the sequence of the protein. We have not yet gotten to the place where we can predict the tertiary structure from the primary sequence. It's an area of very active research, and there are programs that are better than others at it, but no one would claim yet that we can do it for any structure, any sequence that you just hand off randomly. We do know from studying proteins folding that we can see little regions of secondary structures form locally from the hydrogen bonding in that area. And then these little local areas of secondary structure collapse down into this state, the molten globule, because of the hydrophobic collapse. Like if you've shaken up salad dressing, the oil doesn't instantly separate out. It takes a little time, especially if there's a little bit of mustard in there. And what it's doing is it's rearranging and finding that state. Once the molten globule has formed, which is sort of the overall approximate shape, these side chains can then continue to rearrange in smaller ways to come up with that final packing and pattern. And that process of folding can actually be catalyzed. So in other words, a chaperone doesn't change what tertiary structure forms, but it can change how fast it forms. And there are some proteins which we have not succeeded in folding unless there is a chaperone around, which is interesting. And this is then just showing you what can happen. I think I've really basically covered a fair bit of this already. In addition to the primary sequence or primary structure and prosthetic groups like heme groups or B vitamins, some proteins have covalent modifications. They have little places on them where something chemically has actually been covalently bonded to them. This is an example of palmitylation where it has palmitic acid added. We're going to see that that's something that happens to peripheral membrane proteins. Here's a protein that's been glycosylated. I know one protein chemist who claims that everything is modified if you look hard enough. I don't know if that's literally true, but it certainly is true that more things have covalent modifications than we originally guessed at. There's also an increasingly wide range of what those modifications are. It can even include a whole other protein. So, for instance, the protein ubiquitin is a protein in its own right that can be covalently attached to other proteins. These modifications do a couple of things. They can increase the range of properties that proteins have, just as prosthetic groups can. And in some cases, the modifications can regulate things, turn them on or off, or change their affinity for things, or even change what they bind to. Here is one of the more beautiful prosthetic groups. Again, prosthetic group means thing that helps you with things, like a leg prosthesis can replace a leg. And the heme group is called a tetraperol ring. So this group by itself would be a perol, and it's got 4 of them, so it's a tetraperol ring. Typically, we find hemes with an iron in the center, and a very commonly used protein for things that either involve oxygen or that involve electron transfers. We see the heme prosthetic group way, way back. It's one of the very ancient inventions of living systems. Both prokaryotes and eukaryotes use heme proteins. Not the same way. If we look at myoglobin, we have, let me just make sure, okay. If we look at myoglobin, we see that the heme ring is not just sort of floating in space, but it's in a particular niche, a little space that the myoglobin protein has generated that is perfectly adapted for holding that heme so it can do one thing very well. And that is that the heme ring here can bind an oxygen molecule reversibly. Now, the reversibly is the important part. The iron in the heme ring is Fe2. If Fe2 binds oxygen permanently, we call it Fe3 or rust. And so it's an important property of the heme ring in the context of myoglobin that this oxygen binding is reversible. And that is possible partly because there's a histidine on one side of the heme ring that is holding on to the heme iron, and there's another histidine on the side where the oxygen is that's coordinating with the oxygen to help that binding be reversible. The region around this heme ring is called the hydrophobic pocket. That is, when we look at the structure of myoglobin, the residues in the area of the heme ring all have hydrophobic side chains, and so water can't get in there. It protects that iron from interacting with water, which would oxidize it. Any questions? Now, oxygen binding is reversible because of heme, and it's not an all-or-nothing phenomenon. That is, the myoglobin in your body isn't all occupied with oxygen or none of it. Some of it is and some of it isn't. And as a way to talk about oxygen binding, we need a number. And like many of these things that look at affinities, like pKa or like pH or melting temperature, when you have a half saturation, you can talk about a half saturation. The P50 describes that point at which the compound is half saturated. You can see how oxygen binding continues to rise with higher and higher concentrations of oxygen, but it hasn't completely gotten there yet. And it's because these hyperbolic relations tend to taper that it's more useful to talk about midpoints, about half saturation points. And for humans, the P50, the half saturation concentration, is about 2.8 torrs of oxygen. So not a lot of oxygen needed to make it half saturated. Now, hemoglobin, by contrast, is a more complicated protein than myoglobin. Hemoglobin is a heterotetramer, which means a tetramer means there's 4 polypeptides in it, 4 subunits in it, and hetero meaning there's at least 2 kinds of things in it. And these are just showing you some of the other kinds of formations you can talk about. You could talk about a homodimer. If there were 2 subunits but they are the same, that would be a homodimer. If there are 3 subunits, you talk about a trimer, and so on. In general, the subunits within a hemoglobin heterotetramer, or within any quaternary structure, are the same kinds of forces that are operating in tertiary structure. There isn't some magical Velcro that pops into existence for quaternary structure. Quaternary structure, again, to remind you what we mentioned before, is the idea of the overall packing in space of all of the subunits, more than one polypeptide chain. In general, when we look at proteins that have quaternary structure, so these are sort of a little gallery here of other proteins that have quaternary structure. Here's a trimer here. I'm not even going to try and guess how many changed in some of those others. But a common feature for things with quaternary structure is symmetry. They tend to have some kind of symmetry. It may be mirror symmetry, it may be rotational symmetry, but there's usually some kind of a symmetrical property to it. We also commonly find that proteins that have a quaternary structure have one or both of a couple of other characteristics. They usually are larger. You might think that's just because they've got the extra subunits, but you don't tend to find little bitty multimeric proteins. And also, it provides for a new kind of regulation, and we're going to see what that looks like as we go on through the semester and see other examples of quaternary structure and advanced complicated regulation. Now, it's interesting to compare hemoglobin and myoglobin at the different levels. We can see that they're all common descendants of the same ancestor, the ancestral globin molecule. And if we try to compare, as we do here, myoglobin and the alpha and the beta chains of hemoglobin, we can see that there are a number of features that are all going to be the same. They all have a hydrophobic pocket. So here's a hydrophobic pocket in here with a heme group in it with the iron in it. They all have, in the F8, in the F helix, the histidine in the 8th position holds on to the iron 2. And the histidine at the E7 position can hydrogen bond to that oxygen to stabilize it. So those 2 histidine residues are in the same position in all the globins. Those two histidines have an essential function to play, and so structurally they can't be changed. So if you look at these cartoons here of these tertiary structures, you can see that the tertiary structures of myoglobin and hemoglobin are very similar, which makes sense because their functions have a big overlap. All of them have to be able to bind oxygen reversibly. But look at how different their primary sequence is, 18% identical. So over the years, almost anything and everything that could have been changed has been changed between these different globins. So there's clearly more than one way to accomplish that tertiary structure. Many different primary structures can end up at that tertiary structure. So when we talk evolutionarily, we can talk about any given residue in that polypeptide being one of three things. They might be invariant, meaning for that protein family, we always find that residue there. So for instance, His F8, His E7 are examples of invariant residues. They have to be there or they won't work. We can see conserved residues. And typically, conserved residues mean that they've kept the most important property, but they may have changed to another amino acid that shares that property. So a glutamine for an asparagine is a conservative change. A valine for an isoleucine is a conservative change. And you might want to go through your amino acids and ask yourself which of these things would be conservative substitutions. In some cases, it depends on conservative for what. Maybe you just want to make sure it's polar. If all that's important is that it's polar, you're going to have a little wider spectrum of things to pick from than if you want something that's positively charged, for instance. And then you can have some residues that are variable, where a very large number of residues may end up in that position. It's rarely any residue because there is this tendency to have the hydrophobic stuff inside and the hydrophilic stuff outside. But there are positions in which you might find 10, 15 different residues occupying that position. Okay. And so we can take advantage of these changes as a way to trace back the evolution of things. Both the amount of differences, how many of them have changed and what kinds of changes there are, can allow us to generate, for instance, trees that look like this one. And this is a tree that shows you, if we trace back and reconstruct by logic what the earliest globin looked like, we can tell that myoglobin separated quite a long time ago from the hemoglobin globin molecules. And here we have the various globins that we find in hemoglobin. Alpha and beta are what we have in the adult bloodstream. Some of these others are ones that are more common in the fetus, for instance, and have been more recently developed than the other ones. This kind of variant, invariant tracing back and so forth can be very powerful to the point, for example, where it has happened that molecular biologists have taken fish species for which in a number of cases it was known how their immune systems worked, and in some of them it wasn't, generated this kind of tree and deduced that X, Y, and Z species should have this kind of immune system, gone out and checked, and there it is. So it can be a very powerful method indeed, even predictive, not just descriptive. Here you can see just some examples of it. What they've done here is to make sort of a block diagram. Here's the A helix and the B helix. And you can see that there are some residues that are the same in all of them. Here's a tryptophan. It's the same in all of them, and a glycine next to it, and a lysine next to it. And then there are other positions where either it's variable or even missing. So here's one where it's actually a little bit shorter space than the others. You can have actual deletions or insertions. Okay. Now I showed you a curve already of how myoglobin binds oxygen. It's a sort of a simple hyperbolic curve. If we look, here's what we mean by hyperbolic curve, like that. Now, if we look at the oxygen binding of hemoglobin, it's rather different. So here's the oxygen concentration rising along this axis. Here's the fraction of saturation on this axis. And what we see is that with rising oxygen, there's initially a relatively slower rise in saturation, and then a more rapid one that then tapers off again. And we call this shape of curve a sigmoid curve. It's kind of S-shaped, right? Hyperbolic curves are easily described by the simple equation we looked at back with the myoglobin. Sigmoid curves usually involve some kind of cooperative behavior. The P50 for myoglobin was around 2 to 3. The P50 for hemoglobin is around 26. So hemoglobin has a larger P50, therefore a smaller affinity, a lower affinity for oxygen. It takes more oxygen around for it to be half saturated. Okay? But the actual affinity is different depending on how much has already been occupied on that hemoglobin molecule, so that the last oxygens that bind 100 times more tightly than the first ones that are binding down there. So the molecule is changing as the oxygens are binding. And what this allows the body to do is very slick. It means that when the hemoglobin is in the lungs, so the capillary bed of the lungs is exposed to air, the hemoglobin will be fully saturated with oxygen, or very nearly fully saturated. As it leaves the lungs and goes out to the tissues, the concentration of oxygen out in the tissues is quite a bit lower than in the lungs. Because for one thing, it's not in contact with the air, and for another, it's doing work and it's using up the oxygen locally at a steady pace. And so the oxygen concentration in the tissues is between 20 and 40 torr. And now look at how clever the hemoglobin is. The half saturation, the P50 for it, is right in the middle of that range. So it's at a good place to let go of that oxygen. And since the myoglobin has a higher affinity than the hemoglobin, the overall effect in the tissues is for the oxygen to leave the hemoglobin and be transferred to the myoglobin. The myoglobin is only found in the muscle cells. So it's acting as a reservoir of oxygen in the muscle cell itself. Any questions about that before we go on? Okay. The actual structure, the quaternary structure of hemoglobin changes when it binds oxygen. It's a reversible change that happens back and forth each time. So if we look here as a schematic of deoxyhemoglobin, you have an alpha-beta dimer here, an alpha-beta dimer there. Those subunits are going to rotate a little bit around each other and end up closing so there's a smaller cavity in the middle in the oxyhemoglobin. So this is just a very schematic way of representing the fully unoccupied state, or the deoxyhemoglobin, and here's the oxyhemoglobin. And this changes mostly at the tertiary, at the quaternary structure. Myoglobin obviously has to have at least a little bit of a change to let that oxygen in, but it's nothing like as dramatic as the quaternary structure changes of hemoglobin. So there's actually this little cavity here. Keep in mind that each of these subunits has a structure a lot like myoglobin. So there's a hydrophobic pocket here and a hydrophobic pocket there and one here and one there. And then there's this central cavity that's a little different. We're going to see what it's for. Now that's a pretty amazing thing to have happen. And you might ask yourself, how can hemoglobin know that it's going on? How can this kind of change go on? It turns out to be almost a mechanical thing. So the heme ring is a little different when it's occupied with oxygen or not. And so this picture is showing you, this is the deoxy state, and then that's the oxy state up there. And when an oxygen is binding there, it's going to tend to pull that heme ring up, which tends to pull up this F8, this histidine on the F helix. That pulls up the whole F helix because it's part of that helix, and that tends to make the dimer swivel a little bit, and when they swivel, that central cavity gets smaller. These two states are called T and R, or tense and relaxed states. The deoxy state is the tense state, and the oxygenated is the relaxed state. We can even trace these changes down to the level of actual residues along the protein. So if you look really, really closely, you can see how these side chains are sort of sliding, are sort of ratcheting across each other between the oxy and the deoxy state. This is a histidine residue that is going to jump over that threonine to sit there instead. So it was here, and now it's up there. And there are a number of the contacts that can stabilize one or the other state. Sorry, previous, here we go. We call the ability to change the behavior of the thing or the structure of the thing from a site other than what's going on as the primary function, allostery or allosteric. So oxygen binding at one subunit changes the affinity of another subunit. Something binding over here changes the behavior over there as an example of allosteric behavior. Okay? In the case of hemoglobin, some of that allosteric behavior is from one oxygen binding site talking to another oxygen binding site, but there are other allosteric sites in hemoglobin as well that don't bind oxygen and yet can change the behavior of the oxygen binding sites. Now, there are a number of things that can interact with hemoglobin and change its affinity. So deoxyhemoglobin, for example, can bind protons or hydrogen ions. And so you have an interaction that looks something like this. Out in the tissues here, where you have, you're maybe building up some acid because you've been exercising. You talk about lactic acid and so forth. You've got some excesses of acid here. The deoxyhemoglobin can pick up that proton, be carried by the capillaries out towards the lungs, in the lungs, when oxygen binds, it will tend to release the proton, the hydrogen ion, into the blood there, and the hemoglobin will be recharged with an oxygen. That fully charged hemoglobin gets carried out to the tissues, and in the tissues, the presence of the protons helps it to dump off the oxygen. So this sensitivity to pH increases the ability of hemoglobin to ferry oxygen out to the tissues. It's coupled with bicarbonate metabolism. So if you are actively metabolizing, you've generated carbon dioxide. When it becomes hydrated, it can make here's a carbonic anion, and there's the proton that we took out to the lungs. This bicarbonate ion can come out to the periphery. It'll pick up the proton that was released when the hemoglobin was occupied by oxygen, and now you can blow that out as carbon dioxide in your lungs. So this called the Bohr effect helps actually remedy a couple of things at once. It facilitates getting rid of carbon dioxide that's produced during metabolism, and it helps to keep the pH correct. It increases the ability of hemoglobin to deliver oxygen specifically to the tissues that are most active. So it increases the appropriateness of what it's doing. And how big is the Bohr effect? This is showing you a comparison of the sensitivity of hemoglobin at different pHs. Here's myoglobin out here. Its P50 is down around 2 or 3. Again, the lungs with a high concentration of oxygen, the tissues down here. If you have a fairly acid pH, as you might have in very actively metabolizing tissues, look at how large a fraction of the oxygen has been released into that tissue. Whereas if you have another tissue that is actually a bit alkaline, it's not going to release very much of its oxygen in that tissue. So the Bohr effect makes a substantial difference to how large a fraction of the oxygen is released in the tissues as a function of how active they are metabolically. We know enough about the structure of hemoglobin now to explain a very important disease, which is sickle cell anemia. And it's also an example of the power of protein structure and its specificity. Sickle cell trait and sickle cell anemia are genetic traits that are most commonly found in people in or from Africa. And they are the result of a single amino acid substitution. One amino acid substitution in hemoglobin. If you have that substitution from both your mother and your father, so that all your hemoglobin is like that, then you would have what's called the sickle cell anemia, sickle cell condition. If only one of your parents gave it to you, so you're heterozygous for it, you now have sickle cell trait. Either way, the hallmark of the sickle cell gene is these red blood cells that are curled up like that, or sickled. And if you have a double dose, one from each parent, this tendency of the cells to sickle like that means that they can clump up in capillaries and cause a variety of problems in the kidneys and the joints and so on. If you have just one copy, you can detect that that's there, but it often has no other phenotypes. Now, if the double dose is so bad, why is it around in relatively high frequency? Well, it turns out that the single dose, the heterozygote, has a relative resistance to malaria. So the sickle cell hemoglobin is as common as it is because the heterozygotes had a relative protection that the people with the more common variant don't have, and the number of people who actually get sickle cell disease is small enough to balance that out. This is what this looks like at a molecular level. Here's the single amino acid change, and it puts a valine on the surface of the hemoglobin. Now, valine is what? Polar or nonpolar? Nonpolar. So valine is nonpolar. It's a little V-shaped 2-methyl groups, okay? You had a whole snow day to learn these guys. Ah, well. Valine is hydrophobic. It's nonpolar. It doesn't want to be in contact with water. And so that one amino acid substitution on the surface of hemoglobin means that hemoglobin S can make these filaments in which they pack against each other and put that valine out of contact with the water. And because it can make these filaments, they can end up changing the shape of the red blood cells and having all the other consequences. So a good thing to have to the extent that it protects you from malaria. Too much of it, not so good. But in any case, a single amino acid can do the whole thing. Now, I mentioned that there is this cavity here in the middle of hemoglobin. It's distinct from the hydrophobic pockets. So this is a deoxyhemoglobin. You can see heme, 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 4 heme pockets, 4 hydrophobic pockets. And there in the center of it is another space which is called the central cavity of the hemoglobin, or the BPG pocket. Now, the molecule 2,3-bisphosphoglycerate looks like this. So it's got 2 phosphate groups on it. Here it is drawn out in sort of a larger form. So there's there are multiple phosphate groups on it. The phosphates are negatively charged. So you're putting this very negatively charged molecule into the middle of the hemoglobin molecule. And if you look at the BPG pocket there in the middle, you can see various positively charged side chains. So here's a lysine and a histidine, histidine, histidine. Those are all positively charged. There's another lysine there. Those are all positively charged side chains that can interact with this negative charge. And so it can sort of sit inside there, interacting with the hemoglobin. If the BPG is not there, there's all those positive charges that are going to tend to push each other apart, right? So when the BPG is in there, it stabilizes the deoxy state. It makes the hemoglobin state, a more stable state, and it tends to make it easier for oxygen to be released. And this gives you a feeling for how different it would be with or without the BPG. This is the curve we've been looking at so far of hemoglobin, adult hemoglobin with normal levels of BPG. Here is hemoglobin in the absence of BPG. Its curve is much more similar now to myoglobin than it is to hemoglobin. The discovery that BPG could normalize the oxygen binding of hemoglobin was made here at the University of Iowa over 30 years ago, maybe over 40 years ago. It was the discovery that you needed to put glucose into red blood cells to make blood transfusions last more than 2 or 3 days. Red blood cells have no nucleus, they have no protein synthesis. What did they need their glucose for? They needed their glucose to make BPG. And without the BPG, the hemoglobin reverts to being a lot more like myoglobin than like hemoglobin. Other examples of how you can fine-tune the affinity of hemoglobin is that fetal hemoglobin is not the same as the mother's hemoglobin. It has a slightly higher affinity for oxygen than the mom's does, and so the fetus is able to withdraw oxygen from the mother's circulation. And here's another way of representing deoxyhemoglobin, and you can see here in the middle the BPG molecule stabilizing the structure with the positive charges. Out there at the periphery, you can see, I think, all 4 of the heme rings. There's a number of ways to represent protein structures that some are better for one thing than for another. And this is just a chance to show you what's called the sausage diagram, where you don't even have full ribbons, but you represent a helix with a sausage. And that's the most cartoon-like of all, but in some ways also the easiest. And so this is an example here of hemoglobin drawn with a sausage diagram. And you can see the heme rings here held in contact with the 2 histidines. This is trying to emphasize the idea that you can actually think of hemoglobin as a dimer of dimers. You can think of it as a dimer of alpha-beta globin. And so one alpha-beta dimer rotates past the other alpha-beta dimer pretty much as a unit. And this particular sausage diagram is trying to emphasize that dimer within the tetramer aspect of it. The heme ring itself is structurally complex and has to be able to change in response to the chemical environment around it. And this is giving you a sense that heme as a ring can undergo a number of different conformations. I don't expect you to memorize them, but just gives you a sense of how versatile heme can be, that it has anywhere from a shape kind of like this, which looks a little bit like a potato chip, to this that looks a little bit like a Barcalounger. So there's all these different kinds of twists and turns to the side things which turn out to be important in other heme-containing proteins. And then this is another way to sort of represent all of these interactions that we've been talking about to kind of refresh for you that what we're talking about is, the lungs trying to feed the tissues, very typically the muscles, with oxygen, and the different interactions that are going to go on while that happens. So, have a good weekend. Any questions?
This one. Oh, this seems backwards.